Okay, so welcome back to this next video in which we are discussing circadian rhythms. Okay, right, so we've discussed that even if you take a human and put them in isolation where they have no exposure at all to zeitgebers, such as the day-night circadian rhythm, then their body temperature and other physiological parameters will continue to vary on a 24-hour cycle, basically. Okay, and we want to now see how is that maintained. Okay, so it suggests that there is an internal clock, basically, that is regulating these physiological parameters. And we think this physiological clock is the suprachiasmatic nuclei, which are within the hypothalamus of the brain. Okay, so we've discussed the anatomy of the brainstem and the diencephalon, as it's called here, which is this region containing the thalamus, the hypothalamus, and structures around them, such as the optic tracts and things like that. Okay, and we've seen where the suprachiasmatic nuclei are present within the hypothalamus. Okay, one last thing I'd like to say with regards to the anatomy is a discussion of how big these nuclei are. Okay, and also I'd just like to mention what a nucleus actually is, because I don't think I ever defined that. A nucleus is just a collection of cell bodies within the central nervous system. Okay, so when you have collections of cell bodies that are outside of the central nervous system, that's known as a ganglion. When you have a collection of cell bodies within the central nervous system, that's known as a nucleus. So these are just collections of cell bodies, basically. So let's discuss how big the suprachiasmatic nuclei are. Okay, so they're around 0.3 millimeters cubed. So let me tell you what that actually means by drawing you a picture. Okay, so here is 1 centimeter cubed. Okay, so if we want 1 millimeter cubed, we need to take 1 millimeter by 1 millimeter by 1 millimeter, and it would be something tiny like that, basically, in this picture. So a tiny little, 1 millimeter cubed is tiny, but they're even smaller than that. They're 0.3 millimeters cubed. Okay, so take this, divide it up into 10 pieces, take 3 of those, and that's the size of a suprachiasmatic nucleus. So they are very, very small little structures within the hypothalamus. Okay, so that now finishes the anatomy of them. Let's talk about their physiology now, because that's what's interesting. Okay, we think that these are the internal clock. Now, why do we think that? Well, basically, many of their different activities show a 24-hour rhythmic cycle. Okay, so if you take a neuron of the suprachiasmatic nucleus out of the suprachiasmatic nucleus, its behavior shows a circadian rhythm. Okay, so what we can do is plot a graph, one of these graphs, where we plot 24 hours on the x-axis here, so 0, 6, 12, 18, all the way up to 24. Okay, so this is time on the x-axis here. And then now on the y-axis, let's have the activity of the neuron, and I'll tell you what I mean by activity in a moment, because there's many different things that all show this same pattern. So at the moment I'm going to generalize and just say activity, but I'll give you specific things that you can put in place there of activity in a moment. Okay, so what you find is that its activity will be quite low during the night time, and then its activity will go up during the daytime. Okay, and then it will sort of drop back down again during night time. Okay, and then it will repeat this entire thing all over again the next day. Okay, so what sort of activities fluctuate like this within this neuron? Well, for instance, how many action potentials it fires. So the action potential frequency. Okay, and let me make sure that you completely understand what I mean by that. Okay, so if we have time here, okay, now we'll have time in seconds rather than time in hours. So we'll have 0, 1 second, 2 seconds. Then let's mark on this time scale every time the action potential fires, every time the neuron fires an action potential. So So I'll mark it in purple. So let's say it's firing action potentials at roughly about, 1 every second, or a little bit higher than that. Maybe around 4 every 2 seconds, that kind of thing. 1 every half a minute, maybe. Okay, right. 1 every half a second, maybe, around there. Okay. If it started firing them much closer together, okay, so if instead it started looking like this, so it might fire them more and more like that, all in the same time period, so that's what I mean by frequency. I mean how quickly it's firing action potentials, okay, so how many is it firing within a second is what is meant by frequency. So you find that the number of action potentials that the neurons within the suprachiasmatic nuclei fire a second rapidly, hugely increases during the daytime hours and then decreases during the nighttime hours. Okay, so that's one of the things that you could count as activity, basically, the frequency of action potential firing. In addition, also the metabolic activities of these neurons change. Okay, so the famous example is that these neurons also secrete the hormone vasopressin. Okay, and their production of vasopressin hugely increases during the day and then decreases at night. Okay, so vasopressin secretion is another example of an activity measure, basically. So these neurons have this rhythmical level of activity within them that continues even if you remove them out of the suprachiasmatic nucleus. Okay, right. And also, what you can now do is you can ablate these suprachiasmatic nuclei in animals and they lose their regulation of these physiological parameters that occurs in these 24-hour cycles, basically. So we think that the suprachiasmatic nuclei are responsible for the circadian rhythms of these physiological parameters. Okay, so their activity varies on this 24-hour cycle, and then they trigger downstream pathways which then also fluctuate on the 24-hour cycle, and this then leads to these physiological parameters which we can measure also fluctuating on the 24-hour cycle. So we think the suprachiasmatic nuclei are responsible for this, and as I say, if you ablate them in an experimental animal, you can remove the circadian rhythms of these physiological parameters. You don't abolish the animal's sleep-wake pattern because there are different mechanisms that aren't well understood which control the sleep-wake pattern, but as for the physiological parameters, you can destroy them by destroying the suprachiasmatic nuclei. So, how do these nuclei have this fluctuating level of activity on a 24-hour cycle, basically? What's the mechanism by which the suprachiasmatic nuclei neurons have these fluctuations which vary on a 24-hour basis? Well, basically, this involves things known as clock genes, okay, and these have been found within Drosophila. So I'm going to tell you about the ones which are in fruit flies, but similar things are going to exist within humans as well. So, the name of the clock genes within fruit flies are period, timeless, and also clock. So these are names of genes, believe it or not. So you have the period gene, you have the timeless gene, and then you also have the clock gene. Now for short, the period gene is often abbreviated as the PER gene, okay, P-E-R, and the timeless gene is often abbreviated to T-I-M or TIM. Okay, so how do these genes lead to some sort of rhythmical 24 hours fluctuation of activity within a cell? Well, basically what happens is their level of expression varies on a 24-hour basis, and it just works through negative feedback. Okay, so you can set up a 24-hour rhythm very easily, actually. Okay, so let me describe this to you. So let me start by setting up this graph. Okay, so what's going to happen is when let's say you start off with very low levels of one of these clock genes. Okay, so it's right down here. So you've got very little of this protein actually having been made. Okay, so let's say it's period. So let's draw the period protein here. Okay, and you've got very low levels initially, so that's what we're going to plot on the y-axis. We're going to plot period levels. Okay, so we start off with low levels, and what happens is if you have low levels of period, the gene for period, okay, so let's say we've got a period gene here, okay, you will get a large production of mRNA from the period gene. So here is the period gene here. I think I'll add some colour on here. So let's colour in the period protein in turquoise. Okay, so here is the period protein in turquoise. And then we'll have the period gene in blue here. Okay, and of course the period gene can be transcribed into a piece of mRNA. Okay, so here is the period mRNA here. And then the period mRNA can be read by a ribosome to produce period protein. Now, when there is low levels of period, what happens is the period gene will produce a lot of mRNA, so it will produce a lot of period mRNA, and you'll get a lot of period protein being produced. So period protein will start going up, basically. Okay? And after a short delay. So after a short delay, period protein will go up. Then what will happen is, as the period protein accumulates, the period protein can then start to have a negative feedback effect on the actual transcription of the gene, so that you produce now less period protein. Okay, so the accumulation of period protein within the cell stops the production of more period protein. Okay, so you're no longer producing more, and then what's going to happen is gradually the period protein will be degraded within the cell, so the amount will go down, and then as the amount goes down, you will no longer have the negative feedback anymore, so you'll then produce more. Okay, so after a short delay, what will then happen is it will go back up again, and this cycle overall takes 24 hours. So basically just a simple negative feedback mechanism like this, whereby the level of the protein controls the amount of new protein that you're going to produce, you can actually get a fluctuation that cycles on the 24-hour basis. Okay, and these genes which do this are called clock genes, and 3 examples of clock genes which have been found within Drosophila are period, timeless, and clock. Okay, so there will be clock genes like this within the suprachiasmatic neurons, the suprachiasmatic nuclei neurons. Okay, and then they will have this cyclical activity, like so, which will be on a 24-hour basis. And then all we then need is the levels of this period protein, or one of these other clock genes, to determine other activity within the suprachiasmatic nucleus neuron. Okay, and then you can start to get the activity of the suprachiasmatic nucleus neuron being influenced by this, okay, and showing a rhythmical 24-hour cycle. Okay, right, so that's the basis of how we believe the activity of suprachiasmatic nuclei neurons fluctuates on this 24-hour cycle, that you will have clock genes like this. Now, what then happens is the suprachiasmatic nuclei neurons then link to all sorts of other structures. So for instance, they link to other places within the hypothalamus. Okay, so let me show this. So there are many important nuclei within the hypothalamus which control an awful lot of physiology. So the influence of the suprachiasmatic nuclei can then lead to the fluctuations of these physiological parameters on a 24-hour cycle. They also have projections to portions of the midbrain. So by projecting to the hypothalamus and the midbrain, these suprachiasmatic nuclei neurons are then going to influence these physiological parameters, and they're going to result in these physiological parameters varying on the 24-hour cycle, and therefore showing a circadian rhythm. Okay, now, there is one question that remains, however, which is, we need the activity of the suprachiasmatic nuclei to be perfectly in time with the day-night cycle. Okay, so we've now agreed that we have this clock, this internal clock, which has its activity fluctuating on a 24-hour cycle. Okay, but the thing is, we want this peak in the activity of the suprachiasmatic nuclei neurons to be during day time. Okay, we don't want it to be in the middle of night. So why is this clock kept in synchrony with the day-night clock, basically, is the question we have. Okay, we need to know, how do we keep this clock nicely synchronous to the day-night clock? Because you will know that if you move to another country, okay, initially it's very, very difficult because your internal clock will be running not in synchrony with the external clock, but you will adapt very quickly in the upcoming days, okay? And your internal clock will move so that it's in synchrony with the external clock of day-night, okay? So we can change our suprachiasmatic nucleus's phase, basically, okay? So we can change when this clock is actually going to be giving the spike in activity, okay? And we want to look at the mechanism by which light stimuli from the external world, which indicates the time in the external world, how this can influence the suprachiasmatic nucleus's timekeeping. But we'll look at that in the next video.